Myself, Dr. Lectin Mukherjee, Assistant Professor and Head of the Department, Shri P. M. Patel Institute of PG Studies and Research in Science, affiliated to Sardar Patel University, Anand. The title of my today's talk is Lectin Pathway of Complement System. Whereas antigen antibody complexes were required in classical complement pathway. Classical complement pathway started with C1q, which is a complex of C1q, C1r and C1s, 6 into 3, 18 stalk-like molecules of C1qRs, and antigen antibody interaction was necessary. And we stated that classical pathway required immunoglobulin M or immunoglobulin G. Okay. Then came alternative pathway. Unlike classical pathway, just now today's lecture we studied, alternative pathway does not require antigen antibody interaction. So, alternative pathway can be a true replica of innate immune system. Also we studied alternative pathway can be introduced by 3 different modes, that is alternative takeover pathway, alternative properdin activated pathway and alternative protease activated pathway just few minutes back. Now we are going to study the lectin pathway. Again, in lectin pathway the initial steps are going to be different. Rest of the steps, the C2, C4, the membrane attack complex, those steps are going to be similar to classical pathway. So, those steps again we will not be studying, we will just see how lectin pathway is different from the remaining complement pathways. Lectin pathway was very unknown in the initial years. When I say initial years, even after I started teaching, the lectin pathway was very short and sweet. Things were not that prominent because the mechanism was not well understood until a research paper came from National Academy of Sciences by David Heja and Grove. This was in 2012. So, part of my lecture I have taken from this PNAS paper, that is Proceedings of National Academy of Sciences US. This Professor David Heja's paper, he and his group, they revisited the mechanism of complement lectin pathway activation, and this group after a long period of time revealed the role of serine protease MASP1 as an exclusive activator of MASP2. Earlier, even if you read your text, the 8th edition of Kubi, entire glory, the credit, the limelight, everything is given to MASP2, but in fact it is not MASP2, but it is the MASP1 that, according to the research paper, is the exclusive activator of MASP2. So, what I will do, I will just read verbatim the sentence that is given in your book, so that you do not get confused, and then I will compare how the research paper goes slightly different from the conventional understanding that is given in your book. That instead of relying on antibodies, lectin pathway directly uses MBL or mannose binding lectin, MBL. MBL or mannose binding lectin, as per your syllabus, is the first lectin demonstrated to be capable of initiating complement activation, and it recognizes mannose sugar, which is a carbohydrate. This is found exclusively in microorganisms such as Salmonella, Listeria, Neisseria species. Neisseria gonorrhea, you know, it is a sexually transmitted disease it causes, Cryptococcus neoformis, as well as Candida albicans, the fungi or the protozoan. Now, even the membranes of some of the viruses such as HIV-1, human immunodeficiency virus, and respiratory syncytial virus, they have very well to do role with Manan binding lectin. At this point, student might be interested in knowing that if sir is saying that Manan binding lectin or lectin pathway has a role in HIV infection, why people still get HIV infection? Why immune system is unable to clear the HIV infection, especially the lectin pathway? That of course also again I will explain with a research paper. Further, according to your book, in blood, MBL is associated with MBL associated serine protease, that is MASP. What MASP stands for? MASP stands for MBL associated serine protease. MBL associated serine protease. These are the collectin group of families from ficolin, and in blood MBL is associated with MBL associated serine protease. There are 3 MBLs in blood: MASP1, MASP2 and MASP3. Your book mentions that most studies of MASP point to MASP2 protein as the most important player. This is where I want to correct you, right, that according to this research paper, yes, I agree that MASP2 is the major player, but remember serine protease MASP1 is the exclusive activator of MASP2. So, according to this research paper, MASP1 is 60%, MASP2 is 40%, MASP1 auto activates and then it activates MASP2, and together MASP1 and MASP2, just like the classical pathway, it acts on C4 and C2, it breaks C4. C4 is broken down into C4b and C4a. Likewise, C2 is broken down into C2a and C2b. C2a and C4b together forms C4b 2a, that is your C3 convertase of classical as well as lectin pathway, because the same pathway we are going to use. Okay. So, that part of course I am not revising because we have already covered this in classical pathway. Instead, I would go forward. Lectin complement pathway, some of the key points. This is initiated by binding by lectin such as mannose binding lectin or membranes of ficolin family to microbial surface carbohydrates, especially the mannose sugar. The lectin provides recognition function, binding to carbohydrate residue on the microbial surface. Once again, let me revise: classical pathway, antigen antibody complex; alternative pathway, no antigen antibody complex, rather the lipopolysaccharides and other means; lectin pathway, carbohydrate recognition, mannose sugar. The binding activates associated serine protease, that is MASP2. This molecule cleaves C4, just now I showed, it cleaves C4 and C2 to create C3 convertase, just like the classical pathway, that is C4b 2a. So, these are some of the important highlights of lectin pathway. Now, some of the additional points, this is the stock structure of your MBL, Manan binding lectin or mannose binding lectin, and this is MASP1, MASP2 and polysaccharide antigen. So, the lectin pathway is initiated when soluble proteins recognize microbial antigen. Lectins, the mannose binding lectin bind to microbial surface. How it binds? I have just shown the diagram. Lectins can serve as docking site for MBL associated serine protease, that is MASPs. Why docking? Because it is on the surface of the MASPs that the interaction takes place. MASPs in turn cleave C4 and C2 to form C3 convertase. Subsequent steps are the same as classical pathway, and since subsequent steps are same, I am not repeating the lectin pathway in still further greater detail. Now, I just said that HIV-1 also has a role with the lectin pathway, or lectin pathway can even bind to the HIV infection. Okay. This is a paper from nature.com. Okay. This is Professor Wu and group, the good and evil of complement activation in HIV-1 infection. Someone is interested, you can always have this original text research papers from me if you want to read. Otherwise, at least minimum your text you have to follow. This is extra information, of course. So, this paper suggests that complement system is a component of innate immunity, the first line of defense in against HIV-1. Now, what it states, the role of complement system in HIV-1 pathogenesis appears to be multifaceted. That is why it is said that good and evil both. Although the complement system plays critical role in clearing and neutralizing HIV-1 virus, it represents a critical factor for the spread and maintenance. It both clears the virus and it also can help in spreading of the infection. Don't get surprised why it takes place. In addition to complement regulators such as human CD59, vitronectin, which is present in the envelope of HIV-1, these vitronectin CD59 will prevent the complement mediated lysis of HIV. See how the virus is so clever. So, this paper which I have mentioned, this paper reviews the diverse roles of complement HIV in HIV infection. Interesting paper, you can go through. So, what exactly takes place? Very simplified diagram, not like those complicated papers. Viruses, bacteria and other foreign materials are ingested by macrophages. You can see the macrophages ingested the virus, bacteria and foreign materials. After ingestion, the macrophage will stimulate or release chemicals. Those chemicals will stimulate the liver. Liver in turn will release acute phase proteins such as MBP, mannan binding proteins, or MBL, mannan binding lectin, or C-reactive proteins. These proteins, it will give a conformational docking site for the mannose sugar on the bacterial cell wall, antigen antibody complex and the virus envelopes, and this is how the mannan binding lectin or mannose binding lectin it gets initiated. So, all the three pathways, whether it is classical, whether it is alternative or lectin pathway, all three pathways, they will converge at the membrane attack complex. Okay. Here at this point, I would like to give example of Rajdhani Express. Why the train is called as Rajdhani Express? Anyone? So, whether it is classical, whether it is alternative or whether it is lectin, all pathways will ultimately merge at membrane attack complex. This is what we are going to study in this segment in continuation to lectin pathway. Since lectin pathway is very short and sweet, I have included the membrane attack complex along with this segment. So, how membrane attack complex is formed? You have C5, in all pathway we talked about C5. C5 is acted upon by C5 convertase of alternative pathway, C3b, capital B, small b, C3b, or C5 convertase of classical pathway, that is C4b, 2a, 3b. Okay. It will break C5 into C5a, which diffuses away, C5b which remains, C5b initiates the complement components, that is membrane attack complex. How? C5 plus C6 gives C5b6 complex. This binds with C7. C7 undergoes a conformational change. The hydrophobic sites are exposed. After exposure, it will insert into the lipid bilayer. After insertion into the lipid, this is out C5, B, C6, C7. C7 will undergo conformational change. It will bind with C8, and the last player is C9. Once C9 comes, it will form polymerization reaction, and this way a well or kua, the well will be formed. This well will lyse the pathogen, and once the pathogen is lysed, it will insert water or ions and it will kill the pathogen. So, this is the diagram from Janvey and Travers book that I have taken. C5b binds to C6 and C7. It will insert into the lipid layer of the pathogen. Which lipid layer? Not host but pathogen. Within the pathogen, C5b67 complex is formed. This complex will cannot insert into the membrane, right? Once it binds with C8, the C8 complex will insert into the lipid membrane. You can see in the diagram, this will insert into the membrane. Once it inserts into the membrane, then comes the role of C9. C9, can you see the green colored structure? This C9 will bind to the complex and it will polymerize, and after polymerization is complete, it will form this well like structure. This 10 to 16 molecules of C9 MCQ, 10 to 16 molecules of C9, it will form the membrane attack complex. This membrane attack complex is 15 nanometer in height, 3 nanometer it will insert into the membrane, and the diameter of the membrane attack complex is 10 nanometer. This is the artist diagram, and if you see that yes, exactly the same thing takes place or not, you have a electron microscopic diagram. This is how the membrane lesions on the rings, and finally if you zoom it further, magnify it further under electron microscope, this is how the membrane attack complex MAX look inside the pathogen, on the surface of the pathogen. So, this is how membrane attack complex forms. Now, what are the key concepts of lectin pathway? The lectin pathway is initiated by binding lectins such as mannose binding lectin or members of ficolin family to microbial surface of carbohydrates. The lectin pathway provides recognition function, that is recognizing carbohydrate residues on microbial surface. This binding activates an associated serine protease, that is MASP2. Remember, I showed in the research paper MASP2 is in turn activated by MASP1, and this cleaves C4 and C2 to C3 convertase, that is C2A C4B in classical pathway. As for the classical pathway, the end result of initiating enzyme in the lectin pathway is the generation of the enzymes that cleave C3 into C3A and C3B and C5 into C5A and C5B. You all know, students, that C5A is a potent anaphylotoxin capable of generating inflammatory response. Now, some of the questions we will discuss. Okay, once again you can get ready with your chat box. The alternative pathway is activated in response to antigen but does not require antibody interaction. The following proteins are component of alternative pathway except, everything will be component of complement pathway alternative except one, which is not. You can just hit the chat box. I would also like to go with the class option, option A, that is C3 is the right answer. It was a very simple question, but once you get the concepts right, you can always answer. The next one also I just taught in my lecture just few minutes before. certain microorganisms such as Salmonella, Listeria, Neisseria and Cryptococcus neoformis consist of specific carbohydrate moieties on the surface antigen that activate which pathway? Alternative, classical, lectin or all the above pathways. Option C is the right answer, lectin binding pathway, because lectin binding pathway recognizes microorganisms such as not given in the list, even HIV-1 also lectin pathway does recognize. Which of the following component of complement protein enhances inflammation? I just talked, or it is a potent anaphylotoxin. This is a bit tricky because most potent, I want the best answer out of many. It is a uniselect, not multiselect, or it can be a multiselect also. Which of the following component of complement proteins enhances inflammation? It can be multiselect. You want to change the answer, you can. The answer is multiselect. You can change the answer. Okay, Rihan, Pooja, Purvi, Rekha, Neelam Mangarola, yes. The answer is option C, that is both C3 and C5a. But of course, most potent out of this, if the question is modified, which is the most potent, it is going to be option B, C5a. But yes, the question is complement protein enhances inflammation, then of course it is option C. Option C is the right answer, that is C3a and C5a. But someone asks you which is the most potent, the answer would be C5a. So, thank you very much for your attention in the second segment of 25 minutes on lectin pathway.